presenter. My name is Alyssa Jones, and I'm going to be talking to you guys today about how filler selection and lineup construction influences eyewitness identification discriminability. So I think the field generally agrees that fillers should match the witness's description at minimum, but that only gets us to the starting line. So once we get to the point where we agree that description matching matters, the harder question becomes what descriptors should we actually be matching to? This study stems from three related questions. First, are broad general characteristics enough? So Brown et al. And others report that witnesses often provide fairly vague descriptors such as age, race, build, and hair color, while others suggest that witnesses do sometimes provide information about specific facial features. So one question is whether lineups built from these broad descriptors differ from lineups built using more specific featural information. Second, should one type of feature be prioritized? Face processing research distinguishes internal features such as eyes, nose, and mouth from external features such as hair and face shape, but lineup research has not yet fully resolved what that distinction means for filler selection. And third, does description quality itself affect fairness estimates? Prior work has suggested that mock witness fairness estimates may vary depending on the description content that is provided to the mock witnesses. So the broader idea here is that description matching is not just one unitary thing, there are many components. To situate this in the broader literature, lineup construction has traditionally been discussed in terms of match to description and match to suspect approaches. In the match to description procedure, fillers are selected to match the eyewitness's description of the perpetrator. This is the traditional recommendation because lineup members share the described features but vary on the undescribed features. This is what Wells and colleagues refer to as propitious heterogeneity, and this variance across lineup members may facilitate detection of diagnostic features. In match to suspect procedures, fillers are selected to match the suspect's appearance. Historically, the concern has been that matching to the suspect's appearance could lead the lineup to look too similar overall across all the members. But more recent work suggests that the performance may depend also on the structure of the similarity relationships within the lineup itself. The design of the present study included 5 filler selection conditions crossed with target present and target absent lineups. There was 1 target match control condition where fillers matched the target's appearance, and 4 description match conditions. All of the description match conditions started with matching fillers to the broad general characteristics, and then 1 condition stopped there, and the other 3 included additional specific types of features. So 1 condition included additional internal features, 1 condition included additional external features, and then the last condition included additional internal and external features. We did not designate an innocent suspect in the target absent lineups, and we did use a different fillers approach between the target present and target absent lineups. Importantly, all of our lineups were nominally fair based on the Trudow's E standards that are reported in the literature. Our lineups ranged from 4.5 to 5.17. So the question here is not whether fair versus unfair lineups produce different levels of performance. The question is, among lineups that are already deemed fair, what kind of matching procedures lead to lineups that produce the best discriminability? So we expected lineup performance to vary across filler selection conditions. We also expected that description match conditions would differ from one another depending on whether fillers were selected from broad general characteristics alone or from additional internal and external feature information. And more broadly, we expected fairness and discriminability not to map neatly onto one another. So in other words, lineups could all appear fair by standard mock witness metrics and still differ meaningfully in terms of eyewitness performance. Our method entailed 149 participants, first encoding a target face, then completing a short distractor, then making an identification decision from a 6-person simultaneous lineup, followed by them providing their confidence in that decision. They did this for 20 target faces across multiple counterbalance blocks. For anyone in the room who may be less familiar with ROC analyses, an ROC curve plots correct IDs against false IDs across various confidence thresholds. A curve that sits higher and closer to the upper left-hand corner of this ROC space here indicates better discriminability. I use partial area under the curve, or PAUC, as the primary summary measure of discriminability for the most relevant portion of that ROC space. If I collapse all 4 description match conditions together and compare them against target match, the PAUC difference is not significant. So at first glance, it looks like there might not be anything going on here, but this is actually one of the most important findings in the talk because it shows that collapsing all description match based procedures into one category may mask meaningful variations in performance. Once the description match conditions are separated though, a much clearer pattern emerges. There's an upper cluster consisting of the mixed internal plus external condition and the target match condition. Then there's a lower cluster consisting of the broad general characteristics condition and the external only condition. And the internal only condition fell in between these two clusters. This is the real implication of the whole talk. Basically, not all description matched lineups are functionally equivalent, and the content used in the description matching procedure does appear to matter. To summarize the pattern more directly, I conducted a post-hoc comparison between conditions that included lineups created using a mixture of internal and external feature information compared to conditions that used methods where they did not include any kind of internal information. The mixed conditioning was significantly higher in ROC space, but I do want to keep this claim pretty disciplined here. This is a post-hoc result, so I treat it as a strong clue rather than a final conclusion. Interestingly, lineup fairness estimates did vary across filler selection condition, with our two best performing lineup conditions producing significantly lower E values, this is Trudeau's E values, compared to the description matched condition that contained lineups with fillers that were matched to general characteristics, plus additional external feature information, which was one of our lowest performing conditions. However, these lineup fairness differences don't completely explain our discriminability differences due to the general characteristics only condition here, resulting in comparable fairness estimates compared to the best performing lineups. Recently, the broader debate has included a few points relevant to the present study. Lee and colleagues argue that fairness measures are not interchangeable. Relatedly, McKinley and Peterson argue that concepts like propitious heterogeneity and suspect filler similarity have often been pretty loosely defined or subjectively defined. So together, this literature raises the possibility that nominal fairness may not fully capture the structural properties of the similarity relationships within the lineup that may actually matter more for eyewitness performance. And it calls for objective investigations of these similarity relationships. That broader discussion motivated the next part of this presentation, and I want to be clear that this section is exploratory and explanatory, not part of the original causal test. I'm only including it because these questions are a means of helping me describe the behavioral differences in my data. I used an objective similarity approach by utilizing FaceNet, which is a deep face embedding face recognition model, to quantify similarity across lineup numbers as a post-hoc follow-up. The main questions were, what does objective similarity look like in naturalistic filler selection procedures like those used here? Does feature type relate to different similarity structures? And what do these objective measures look like in relation to conditions varying in discriminability and performance? This slide starts to unpack those questions for target present lineups. Mean target filler similarity is relatively flat across conditions, which can be seen here in the left-hand figure, which suggests that one condition is not simply producing fillers that are globally much closer or farther from the target on average. What varies more is the spread of those similarity values, which you can see here in the right-hand figure. The internal only condition actually shows the most even spread, while the broad general characteristics and external only conditions show more uneven spread. So for these lineups, the ones with the most uneven spread, there may be more cases in which 2 or 3 lineup members are more similar to each other than the other 3 or 4 members. This figure links that spread measure to discriminability for target present lineups. More uneven target filler spread was associated with lower discriminability. The broad general characteristics and external only conditions had the greatest spread and the lowest PAUC values, while the target matched and internal external condition had more even spread and the highest PAUC values. But the internal only condition is an important exception. Even though it had relatively even spread, discriminability was only intermediate. This is important because it tells us that spread alone maybe doesn't explain the performance. So the distributions of lineup similarity relationships could be informative, but it may not be the whole story. The type of feature information used to select fillers may matter above and beyond spread itself. Since target absent lineups did not have a designated innocent suspect, I looked at average filler similarity, represented by the left-hand figure, which does not appear to relate to discriminability. And by contrast, fillers stand out, meaning the extent to which some fillers are structurally more distinct relative to others did track the data better. So again, average similarity looks like a fairly blunt tool, but it may be worth investigating whether the evenness or unevenness of similarity relationships within the lineup lead to differences in identification performance. My overall findings fit with recent developments in the literature across studies by Koloff et al., Shen et al., Harris et al., and Toretti and Penrod show an emerging picture that indicates that more similarity is not always better, but low similarity doesn't always seem to be better either. My contribution here is a bit more specific. Within description match procedures, changing the feature content used to select fillers can change the resulting behavior. And it's worth investigating what the content used does to the similarity structure within the lineup itself. There are several limitations here, but they do point us pretty directly to the next study in this line of research. Some of the key comparisons were post hoc and need pre-registered replication. There is no designated innocent suspect, which limits what I can conclude about innocent suspect protection. The FaceNet analyses were exploratory and need further investigation that is pre-registered. And then filler selection involved a single research assistant, which could introduce possible rate of variance. So the next step is a direct pre-registered manipulation of mixed features filler selection conditions compared to external general characteristics filler selection. And then I would like to use human subjects to get similarity ratings, use a designated innocent suspect, and multiple filler selectors per condition. But the take-home message from my study has 3 parts. First, not all description match lineups are equal. Second, in this data set, the mixed internal plus external condition produced the strongest description-based performance, while broad general characteristics and external-only conditions performed the worst. And third, fairness did not map cleanly onto discriminability. Description content did drive differences in fairness estimates, but they didn't completely map onto the observed data. But the practical implication here is just basically that the specific feature content used to select fillers can have a direct influence on identification outcomes. And I would like to say thank you to you all, as well as my lab and my current PhD student, Alyssa Allen. And I just want to let everyone know that I am recruiting a second PhD student. So if you know anybody or are a student yourself that is looking for a position, come talk to me. Thank you.